Hi everyone, my name is Kristina Joe, a mechanical engineering student at San Francisco State University. This video is for the spring 2022 Biomechanics class taught by Dr. Azadi, and today I will be talking about biotensegrities and their influence on the structure of cells. So I'm first going to go over a quick outline of this video. I'm going to first explain tensegrities before going into depth about biotensegrities. Next, I will quickly go over the basics of a cell structure and then introduce the tensegrity model for cytoskeletons. Then I will talk about how mechanical forces influence cell's behavior and how that study eventually led to the creation of organs on chips. So what is a tensegrity? A tensegrity is a structural principle that applies discontinuous compression to disconnected elements using continuous tensile force. Buckminster Fuller coined the term back in the 1920s, noting the structure's integrity and tensile force, hence tensegrity, tensional integrity. The figure on the right displays a very basic tensegrity structure. The green rods are not touching each other, but are in compression due to the red cables pulling both ends of the rod, keeping them in place. The cables themselves are in continuous tension due to the rods pushing and stretching out the cables. Although tensegrities may look unstable, they are in fact incredibly resilient. Tensegrities can be compressed and deformed, but will reverse back to its original shape. This is due to the structure's prestress. Prestress is an equilibrium concept where the structure itself is under stress without any outside influences, or self-stressed. Tensegrities are also modular. For example, the two basic structures on the right can be stacked on top of each other to create a taller, more complex tensegrity. The prestress and modularity of tensegrities allow them to be useful in many applications, including home furniture, artistic sculptures, and even practical applications. The left picture shows the famous 1,570-foot-long Kurilpa Bridge in Brisbane, Australia, and it's an example of tensegrities being used in a real-life practical application. So now this leads into biotensegrities, and biotensegrities are a specific type of tensegrity. The concept is using the principle of a tensegrity to explain structures within the human body. For example, our bones do not touch one another, instead they are floating within our muscles and connective tissue. This is an example of a tensegrity. If we assume that the human body has tensegrity-like structures, then we can also assume that the human body will behave like tensegrity structures. The picture on the right shows a tensegrity model of the spine. Models like this can help us understand the biomechanics of the human body, how the body moves, and what it can withstand. Now that we understand the basics of tensegrities and biotensegrities, we are now going to focus on applying the tensegrity model to cytoskeletons. But first, let's quickly go over the basics of the cellular structure. The idea of the cell structure has evolved over time. Originally, the cell structure was considered just a sac that held fluid and floating particles. Now, the cell structure is more complex. The picture on the right shows a very simplified diagram of a cell. Currently, the structure of the cell is generalized into 4 parts: the cell membrane, the nucleus, the cytoplasm, and the cytoplasmic organelles. The cell membrane is what encloses the cell, maintains the integrity of the cell, and controls what goes in and out of the cell. The nucleus is the quote-quote brain of the cell, or controls how the cell will function and its job. It also provides the basic structure of the cell. Next, the cytoplasm is the gel-like fluid inside of the cell, which keeps the organelles within it in place. It is also where cell expansion, growth, and replication take place. Lastly, the cytoplasmic organelles are the smaller quote-quote organs within the cytoplasm that perform specific roles and functions within the cell. For example, the mitochondrion and ribosomes are examples of cytoplasmic organelles. In the book Cytoskeletal Mechanics, Models and Measurements, there is a chapter called Models of Cytoskeletal Mechanics Based on Tensegrity, where Dmitry Stemanovic writes about how the tensegrity model can be applied to cell structures. He suggests the cytoskeletons remain their shape like tensegrities, with microtubes in compression and extracellular adhesions in tension, allowing the cells to withstand dynamic loads. As seen in the picture on the left, it's a simple example of the tensegrity model being applied to a cytoskeleton. The lines in red are the microtubes in compression, and the extracellular adhesions that are in tension are in green. The tensegrity model contrasts with the contractile stress model, or the idea that cells maintain their shape under contractile stress. The contractile stress model assumes that the cytoskeletons are in a static network, which is not the case in reality. Also, a main feature of a tensegrity is its resilience because of its prestress, which can be used to bring some understanding to how cytoskeletons keep its shape under the dynamic deformality of the cell. Overall, the tensegrity model allows a deeper explanation for complex cellular behavior, and the tensegrity model also allows for cytoskeletons to be simplified to simpler structural models. Don Engr, an American biologist, supports the tensegrity theory in his TED Talk, creating scientific marvels that are works of art. He talks about how cells in a petri dish act like a tensegrity. If you squish a tensegrity, its prestress will return the tensegrity to its original shape. Cells in a petri dish have the same kind of behavior. He also explains that cells have a molecular skeleton that is like our muscles, which is also an example of a tensegrity structure, but also relates back to the biotensegrity model for cellular structures. Engr also found that mechanical forces also influence the cell's structure as much as the chemical makeup of the cell. One of his studies stretched different shaped cells to find how they behaved. He found that flat, pancake-shaped cells grew under stress forces, while rounder cells would die under the same stimulation. Having a deeper understanding of how cells behave helps us use biology as a means of engineering innovation. Engr was able to create human organs onto chips like the lungs, airway, and intestines because he was able to replicate the mechanical behavior of the organs. The picture on the top right is what the lung chip looks like and its size. The picture below shows the cross-section of the lung chip. The vacuum slots on the side create the mechanical forces that are similar to the alveoli in the human lungs. He then injected the chips with cells from the alveoli, like the lung and capillary cells. The chips can then be connected to tubes that would stimulate blood flow. Using these chips has allowed scientists to observe how organs behave on a cellular level while also observing the mechanical forces within the human body. These chips are also used to help advance the medical industry. 70% of medical drugs fail when they reach human trials, and this can be changed using Engr's organ chips. Without putting human or animal lives in danger, drugs can be accurately tested to see how human bodies react to them. For example, different chips with different demographics can be made for the same organ, like the lung. A lung with asthma can react differently to a drug than, say, a healthy lung or an elderly lung. Overall, tensegrities are essentially the structures of life. They are structures that are in continuous tension and discontinuous compression. This leads them to be resilient, modular, and very useful. They can be used in technology and engineering in practical ways and can even be seen in nature through biotensegrities. Examples of biotensegrities can be found throughout the human body, from the skeletal and muscular structure all the way down to the cellular structure. The tensegrity model can be applied to cytoskeletons to understand how they behave under dynamic loads that deform their shape. Biotensegrities even allow us to see how mechanical loads change the chemical behavior of a cell and even create organs onto chips to use in the medical industry. Overall, tensegrities and biotensegrities offers insight to complex behaviors found in nature, allowing technology to apply to nature and nature to inspire technology. Thank you so much for listening. If you would like to reach out to me, my email and LinkedIn are shown here and also in the description below. Again, thank you so much and hope you have a good day.